How's it going, guys? So the opening video for this video actually has to do with what we're learning today. So don't skip it. Just watch it. You'll enjoy it. You'll see what I mean here in a second.
Listen tomorrow night for the concluding adventure of the Black Pirate Ship. Now it's time for Annie's secret message for you members of the Secret Circle. Remember, kids, only members of Annie's Secret Circle can decode Annie's secret message. Remember, Annie is depending on you. Set your pins to B2. Here is the message. 12, 11,
I am
2,
in my first
8,
secret meeting.
25, 14, 11, 18, 16.
Oh, Pierre was in great
23,
voice tonight. I
12,
could tell that tonight's
23,
message was really
21,
important.
3, 25. That's a message from Annie herself. Remember, don't tell anyone.
90 seconds later, I'm in the only room in the house where a boy of 9 could sit in privacy and decode. Aha, B. I went to the next. E. The first word is B. S. It was coming easier now. U. 25. That's R.
I'll be right there, Ma! Gee whiz.
T. O. Be sure to. Be sure to what? What was little orphan Annie trying to say? Be sure to what?
Granny has got to go. Will you please come out? All right, Ma. I'll be right out.
I was getting closer now. The tension was terrible. What was it? The fate of the planet may hang in the balance.
No, Granny's gotta go. I'll be right out. For crying out loud.
Gee, almost there. My fingers flew. My mind was a steel trap. Every pore vibrated. It was almost clear. Yes, 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 yes.
Be sure to drink your Ovaltine. Ovaltine? A crummy commercial?
So today we're talking about the final process in the central dogma of biology. You'll remember that when we talked about the central dogma of biology, it's this idea that your information is in your cells in the form of DNA, but that DNA is then converted or copied into RNA, and then that RNA is used to build proteins, and the proteins are actually the things that give you your traits. So today we're talking about translation, which is that final step of going from RNA and using it to construct proteins. And the video in the cold open here of my video was from A Christmas Story, and he was decoding a code. So I want to ask a question as an analogy here. How do codes work, right? So I have a series of numbers here, and when you're looking at these numbers, they probably just look meaningless, right? When you look at this, it seems to be a completely different language. And the reason why is because it kind of is a different language, right? Codes, languages, things like that, they're not so different. They have meaning, they have characters and symbols and things like that. But the thing to understand here is that this code that was broadcast over the radio in the movie is kind of like its own language. And my question is based on this, right now, can you figure out what the language means? What translate it into English for me? Go ahead. You can pause the video, translate this into English for me. And most of you are probably thinking, well, this is not possible. And why is it not possible? Because I don't really know the relationship between this code and the English language. Now, if you watched my video from the beginning, you'll know that this code actually means be sure to drink your Ovaltine. But can you see how this is such a tough ask for me to ask you to do, right? What is the relationship here between this code and this English language, right? We need something to help us interpret this language and translate it into this language. Now, in the video that I showed you, the main character was using something called a decoder ring. Okay, and so the decoder ring, this is actually from the movie. Okay, the decoder ring was something that they would give to kids in like the 1940s and 50s. And if you look really closely, you can see that on one side of the ring are some letters of the English alphabet. On the other side are some numbers. So you'll see here that R, the dot, when you set the dial to R25, that means 25 is R. Or that P in this case is 26. And things like that, right? Or here you can't see this part, but B is 12, right? So this sort of decoder ring serves as a bridge between this language and this language. And so you have some way of interpreting it by using this decoder ring. Now, I want to relate this to the central dogma of biology. Like I said before, the central dogma of biology is that DNA is converted into RNA and then into protein. And DNA, remember, is what has your information for making you you, but your protein is actually what gives you your traits. So, in a sense here, we have two separate languages. We have a DNA language and we have a protein language. Or we have I guess more accurately, we would say we have a nucleic acid, because both DNA and RNA are nucleic acids. But we're going from a nucleic acid language to a protein language. We're almost translating from one language to another. Now, I want to remind you a little bit about proteins, right? Proteins are the little machines in your body and in your cells that actually perform the tasks at hand. Proteins are made up of monomers called amino acids, and when you string those amino acids together, they make a protein. That is the polymer. Now, the protein, depending upon the order or on the sequence of amino acids here, will bend and fold into a particular shape. So this is a picture that I think's I just love protein structure, but these are different proteins, and so you'll see that they're quite colorful and things like that, but they have different shapes. Some of them are spiraled, some of them are hollow on the inside. When you chain all of these amino acids together, they form a protein that folds into a complex shape. And it's the shape of the protein that gives it its function. In a previous video, I said that proteins are just like tools in a tool set. A hammer has the function that it has because of its shape. A Phillips head screwdriver has the particular function that it has because of its shape. So all of these things we want to keep in mind as we go into this discussion of translation. But what we're focusing on here today is this step. How do we go from the information in RNA to putting that information into protein? Well, this happens at a place called the ribosome. And so we're inside the cell here. And the ribosome is a big, huge molecule. And what it does is it reads the mRNA. Here's our mRNA here. It reads the mRNA and uses that information in the mRNA to make a protein. So these are like the amino acids that are chained together to make the protein. Okay. Now, what happens is that the mRNA leaves the nucleus, right? The RNA is made in the nucleus, right? But then it leaves the nucleus and it goes outside the nucleus. This is called the cytoplasm. And the cytoplasm is where the ribosomes are. So what happens is that the ribosome actually kind of attaches itself to the mRNA. And then what it does is it pulls the mRNA through the ribosome and it reads the mRNA, kind of like how you might read a book or something like that, right? It reads the mRNA, but here's something I want to point out, and this is extremely important, okay? Notice here that it reads the mRNA 3 letters at a time. Okay? And these 3 letters, a group of 3 letters on an mRNA strand, is called a codon. Okay, and the codon's up here. Codon is a group of 3 mRNA letters, and the thing is that those 3 letters code for a single amino acid. There are about 20 different amino acids that are used to make proteins in human cells. So that 3-letter code, that 3-letter codon, is what symbolizes or codes for a particular amino acid. So like here, this codon, UUU, is specifically referring to this amino acid that's right here. Okay, so what the mRNA does is it reads what the ribosome does is it reads the mRNA 3 letters at a time, and those 3 letters, called the codon, code for a particular amino acid. Now, I want to relate this to the genetic code, okay, the decoder ring. So you might have heard in popular culture or something like that, something called the genetic code. Well, what do scientists mean when they say the genetic code? What they mean is they mean life's decoder ring. Because keep this in mind, mRNA is made up of letters like A, U, G, C, stuff like that. It is its own type of language. And protein is made up of amino acids called methionine, isoleucine, serine, things like that. They are completely different languages. Now, it's at this point that I'm going to geek out a little bit. As a scientist, as a science teacher, I'm going to geek out a little bit because it absolutely blows my mind that life, right? Life, the thing that has been around on Earth for about 3 billion years, is dependent at its very core on information. Information stored in DNA and RNA and in proteins. And not only that, this information is very, very, very similar to a language. Now, that fact by itself, to me, is mind-boggling, that living things are almost programmed like computers are programmed, or like how apps or cell phones are programmed. They're reliant on information in order to operate. That is something that's amazing to me. But what's even more amazing to me is that not only is life dependent on one type of information language, it's actually dependent on 2 types of information language, and these 2 are completely separate from one another, right? There's really nothing about this language that makes this protein language necessary. So that's, to me, mind-boggling. But then we pull in the decoder ring. So the decoder ring, this is the genetic code in the middle. So what this is this is called a codon chart. And so what it does is it allows you to read the 3 letters on mRNA and then determine what amino acid that codes for. So for example, let's take AUG, right? AUG. What if I wanted to know, what does that code for? What protein or what amino acid does that code for? Well, I would go to the first letter, A, and then for second letter, I'd go to U, so here, and then I look at AUG, and I see that it codes for methionine, M-E-T. Okay, so this is like our decoder ring that the little boy used in A Christmas Story, right? This tells us how to take this language and translate it into a protein language. Now, here's another thing that's mind-blowing. Okay, as scientists, we have no idea why this code is the way that it is. Like, for example, why does UUU code for phenylalanine? Why doesn't UUU code for leucine or proline? right? There's no answer to this question. We have no idea. And there doesn't seem to be any physical law or law of nature or anything like that that makes this code the way it is. We have no idea why, for example, histine is coded for by the letters CAU and not some other letters. We have no idea. No idea. And so this is, I mean, I'm done geeking out now, but this is just amazing to me. There's just, life is nothing like we thought it was 100 years ago. Nothing at all like we thought it was. It's so much more complex and interesting than anybody could have imagined. So the genetic code here is the relationship between the 3-letter codons in mRNA and the amino acids that they code for. So I'm going to teach you a skill here. I want to teach you how to take DNA code and figure out what type of protein it codes for. So here's an example. Let's say I give you DNA here. And the sequence of DNA here is ATG, CAA, TTG. All right, well, the first thing I want to do is I want to transcribe the DNA into RNA. So if the other side were made of RNA, what would it be? Well, the first one would be U, right? Because A matches up with U in RNA. Across from T, we would have A. Then we would have C, G, U, U, and then A, C. All right, so that is what our RNA would be. What we did is we just transcribed the DNA into RNA. But what I'm going to teach you how to do today is the process of translation. Okay, we're going to take this RNA and we're going to translate it into the amino acid sequence. So the first thing I want to do here is I need to look at things in groups of 3. Remember how I said that mRNA came in letters of 3? Because every group of 3 letters codes for a different amino acid. And so I'm going to figure out what amino acid does this code for. Let's figure it out. So let's start with UAC. UAC. What amino acid would that code for? Well, I need to look at my codon chart. So I'm going to start with my first letter. And once again, it was UAC. Right? Yep. Okay. So we start with the first letter, U. Then the second letter is A. And then under here, I would go down to C. So I would see that UAC codes for tyrosine. So I would put TYR. That's the first amino acid in the protein, is tyrosine. All right, what about GUU? Okay, so my first letter is G, so I'd go down to G. And then my next letter is U, so I would go to the U column. And then it's GUU. So right here I see that GUU is valine. Okay, and then the last one is AAC. Okay, so I start on the first letter here, A, then I go to the second letter, A, then the last letter is C. This is called asparagine or asparagine. So AAC is asparagine, so I'm going to go ASP. So this sequence of DNA is transcribed into RNA, and then the RNA is read 3 bases at a time. And every 3 bases codes for a specific amino acid. And then these amino acids, when we put them together, they form a protein. So this is how we go from DNA to RNA to protein. All right, let's do another example. Okay, so we have DNA here that is TTT, CGG, ACC. So let's turn this into RNA. So what goes across from T in RNA would be A, A. Across from C is G, and then C, C. Across from A is U, remember that, G, G. Okay, so now at this point, we would need to take our RNA and split it into our codons. So there's 3 there. 3 letters codon there. Then another one there. Okay. So at this point, we can use our codon chart to figure out what proteins or what amino acids this RNA codes for. Now, I'm going to use a different codon chart this time, and this is my favorite type of codon chart. I think this is the easiest type of codon chart to use. So the way that you use this is the first letter is in the middle. The second letter is on the little bit outside, and then the last letter is out here. So let me give you an example. So the first one is AAA. Okay, so in order to figure out what amino acid it codes for, I would go A for the first letter, A for the second letter, and then A for the third. And I see that it is lysine. So the first amino acid is lysine. The next one is GCC. Okay, so I would start with the first letter, G, then I would go C, C, it's alanine. Right there. And that is abbreviated ALA. And the next one is UGG. And so UG, so the first letter is U, then I would go to G, then to G. It's a amino acid called tryptophan. We write that TRY. Okay, so this DNA sequence ultimately codes for a protein, and the protein is made by linking lysine, alanine, and tryptophan together into one chain. All right, so like I said, I personally like this type of codon chart. You can use whatever codon chart you want. We're going to post a couple with the assignment this week. You can choose which one you like better. I personally think this one is a lot better. It's just easier to use, right? You just go G, 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 oh, that's glycine, or UCC, that's serine, stuff like that. Okay, so this week for your assignment, what you're going to do is we're going to give you an assignment, and it has background information on how the coronavirus works. The coronavirus has a protein on its outside called the spike protein, and that spike protein is kind of like a key. And the key here is used to gain entry into a human cell. That's what's down here. Now, the way that it gains entry is that the spike protein key just happens to fit into a receptor on human cells called ACE2. So ACE2 is kind of like the lock. And so when the spike protein fits into the receptor called ACE2, the cell actually gets tricked into letting the virus in. Okay, so what I do here is I give you the DNA sequence for the spike protein on the human version of the coronavirus, and I also give you the spike protein for the bat coronavirus. And so what you need to do is you need to take the DNA, convert it into mRNA, and then take the mRNA and use the codon chart to find the protein sequence for both, for the human coronavirus and for the bat coronavirus. After that, you'll have some questions to answer, and then we have kind of an analysis challenge question for you. And what this is this is an experiment done by some scientists. It says a team of scientists performed an investigation in order to find out how strongly the coronavirus spike protein binds to the human ACE2 receptor. So we're seeing how strongly do those two things bind together. So what they did is they created several different solutions, each containing a different concentration of human coronavirus spike protein. That's what this is down here, spike protein concentration. So like down here, these are solutions with or mixtures with a lot of spike protein in them, these are mixtures with not a lot of spike protein in them. So they did this for the human coronavirus spike protein, and then they poured it over a plate, and on that plate was a bunch of human ACE2 receptors. And what they did is they measured how strongly the spike protein stuck to the ACE2 receptor plate. And so here you can see that the bat coronavirus is not quite as strongly sticking to the ACE2 protein or receptor as the human coronavirus, which makes sense, right? So after this, there's a question that you guys need to figure out. That's it for this video. Let Mrs. Cook or I know if you have any questions. Be sure to join us on Zoom at least one of the days per week, Tuesday or Thursday. We'll love to help you out and get some of those assignments done in class. See ya.